A good morning or afternoon to you. Thank you for joining today's webinar. I'm Jan Reichert, Executive Director of the Antibody Society. I'm here with my colleague,
Silvia Crescioli from the Antibody Discovery and Cancer Immunotherapy Group at King's College London.
We will be your moderators. This is the second in a two-part webinar series on fundamentals of the immune system. These webinars are designed to inform and educate our members in the broader scientific community about topics relating to the adaptive immune receptor repertoire. The Antibody Society is pleased to include a group that works actively in this area. They comprise the adaptive immune receptor repertoire community, also called the AIR community. Our speaker today is a very active and long-standing member of the AIR community. Dr. Scott is Professor Emerita of Simon Fraser University in Canada. Today, she will tell us all about the immune system in action. Please note, this webinar is being recorded. The agenda as well as the slides for today's webinar can be downloaded from the Materials tab in the viewer. We will have a question and answer period after the presentation, so please do add any and all of your questions to the Q&A box in the viewer. Without further ado, I'll now turn the show over to Dr. Scott.
Thank you. Thanks very much, Jan. Good morning and afternoon and maybe even evening to you all. In part one, a few weeks ago, I didn't get completely finished with part one. So I'm going to go back and finish chapter 3 and then take you through chapter 4, having to do with orchestration of the immune response. Chapter 3 was kind of setting up the immune system in all its little parts and how its separate little parts function. And then in part 4, chapter 4, we'll be going through orchestration of the responses, putting it all together. Yeah, so where we had ended up was sort of toward the beginning of chapter 3, section B on signaling, activation, proliferation, and differentiation of B cells. We'd already gone through all that for the T cells, the CD4 and CD8 T cells. And at this point then, we were doing it for the B cells, B cell responses. So there are different phases in rolling out a humoral or antibody based immune response. And first off is you'll have these B cells that will have, when they're naive, in other words, they've come out of the bone marrow, they've matured typically in the spleen, and once they've matured, they will have IgM and IgD on their surface as receptors, and they will bind to antigen, and along with the proper co-stimulation, depending on what kind of B cell this is, whether it's a more innate B1 B cell or a more innate marginal zone B cell versus a follicular B cells. There's different subsets of B cells. They'll bind to antigen and become activated. They could become activated either through the help of a helper T cell or also other stimulated, such as LPS that will bind to toll-like receptors and activate the B cells and so forth. Anyway, once you get an activated B cell, which has definitely got a signal through its antigen receptor, it will then proliferate. And especially if it's a follicular B cell that is being helped by helper T cells, it will go through a germinal center response where not only will it develop plasma cells that will secrete antibodies whose receptor is exactly the same as the receptor on the B cell. So now that receptor's turned into a secreted antibody, but with the help of T follicular helper cells, these antibodies, these B cells will also undergo isotype switching, so they'll switch their heavy chain constant region from, for instance, a mu to a gamma. So now it'll retain the same light chain, but switch the heavy chain constant region. That's called isotype switching. So now you will have B cells, plasma cells that secrete IgG or IgA or IgE. Another thing that will happen is that in a germinal center reaction, you will get affinity maturation, where weak binding or the weaker binding B cells will drop out, and B cells with the higher affinity higher affinity antibodies will predominate, and therefore the overall serum antibody response will have a higher overall affinity or avidity for the antigen. And then finally, also memory B cells will differentiate out of that, which will then circulate through the secondary lymphoid tissues, so the lymph nodes and the spleen, etc., looking for the very same antigen to go through this whole process again, essentially. So these are the follicular B cells that are doing this, and these are B cells. They're called follicular B cells because they home to the follicle of the secondary lymphoid tissues, and if they find a T cell that will work with it, then they will form a germinal center response. So I'm just going back a little bit to the origin of these germinal center responses. And the follicle is in the kind of the cortex area, so it's toward the outer area of a lymph node. This is supposed to be a lymph node here, this big thing. This is just a section of it. And the follicle is actually defined to some degree by a network of cells called follicular dendritic cells. They're not the same thing as what we've been talking about, antigen-presenting dendritic cells for driving T cell responses. These are follicular dendritic cells. They don't move around, but they do capture antigen, and the antigen can come to them either from the periphery coming in through the afferent lymphatics. So that's, remember, you've got a flow of lymph fluid coming through and percolating through these lymph nodes on their way back to the right heart, where all lymphatic fluid joins up with basically the superior vena cava going into the right heart. So you've got this movement of fluid getting pushed or pulled, I guess, sucked toward the right heart by the heart. And so these antigens can then flow over these follicular dendritic cells, which can capture it. Also, in this subcapsular space of the lymph node, you have these subcapsular sinus macrophages, which are specialized macrophages that can grab antigen and actually help get it shuttled over. It'll pass it on to what we call bystander B cells that will bring it over to the follicle, weirdly enough. And then also larger antigens can get captured on dendritic cells in the further in the toward the paracortex. And these dendritic cells can pick it up. These are actual dendritic cells that can also activate T cells. And they can shuttle antigen to these follicular dendritic cells. So that's pretty much how antigen can get delivered. And here's just, this is just some staining showing where the follicular dendritic cells are. And here again is the outer part of a lymph node. And you can see staining of radiolabeled antigen there. And the follicular dendritic cells are called that because they have these long dendrites. And they can also pick up antigen that has complement bound to it, complement fragments that have been activated by antibodies and in other ways. There are also FC receptors, so you can actually have antibodies also grabbing antigen on these follicular dendritic cells. And these immune complexes can be sitting around here for a really long time, but they will be getting picked off, these antigens, by B cells, so they can eat this antigen, and if there's any protein in there, process it and present to follicular helper T cells. Okay. Yeah, so this is where we end. That's why I put end here. This is where we ended last. And again, I apologize for not getting through all my slides last time. So this is now a B cell getting activated to signal by virtue of antigen. And this is a weird looking microbe, if you ask me. But anyway, typically a naive B cell, as I mentioned before, will have IgM and IgD on its surface, and along with some other cell surface molecules like Ig alpha and beta, you form a receptor complex, and associated with that is going to be a complement receptor and this whole little apparatus here. So antigen can bind to the surface antibody, and if complement is around, then you can also signal through this complex here. And this is called the CD19 complex because of this associated molecule here that people recognize with antibodies when they're trying to identify B cells. Anyway, so that can stimulate or get going the signaling through the B cell, and of course other IgM molecules and other complement receptor complexes can be recruited into this to form a kind of like a big synapse almost between multiple antigens and the B cell receptor complex. So I don't want to go through all this, but there's an ornate signaling complex that's going to end up activating multiple transcription factors, and just pretty much the exact same endpoint in some ways as a T cell. So NF-kappa B, NFAT, and AP1. I won't go through all this, but the point is that you're getting cross-linking by antigen. And remember, with signaling, we always have signal 1, signal 2, and signal 3. So in this case, with instead of a T cell receptor, it's the B cell receptor for signal 1. Signal 2 is going to be provided by CD40, and which is going to be on a T helper cell. So CD40 is a signaling molecule on B cells, and CD40 ligand on the T cells can help to signal. So that's your secondary stimulation. Plus you have the CD19 complex helping with that. That's over here. And then finally, you also can have cytokines. So signal 3 telling the B cell basically what kind of function it's going to be taking on in terms of isotype switching, because that's pretty much the different antibody isotypes dictate what the outcome is going to be for the humoral immune response. And also, I just want to point out that also you have molecules that have these ITIMs on them. Remember, we have these activating markers on the B cell receptor and T cell receptor complexes that allow, when they get phosphorylated, allow this whole thing to get kicked off. But the ITIMs, which are, for instance, on Fc gamma receptor 2b, so this means IgGs can bind to FcR2b, and they will actually initiate inhibition, so they'll drag in dephosphorylases that will dephosphorylate all this stuff, and therefore shut down the signaling. Excuse me. So, excuse me. So there are mechanisms for activating the signaling and also for shutting it down. So then the sequence of events are a little bit complicated. That's why we do the T cell part first, because T cells are involved in the germinal center reaction. So it's again, you want to think about your protein antigen as having B cell epitopes on their surface and T cell epitopes just about anywhere in the molecule, whereas the T cell epitope is eventually going to be a peptide that has the right anchor residues to bind to an MHC molecule made by the B cell. So the B cell is an antigen presenting cell. There's 3 kinds of antigen presenting cells, which means they have class 2 MHC on them. There are dendritic cells that initiate the immune response. And then there are B cells and also macrophages that are secondarily will display class 2 MHC to help push immune responses. All right, so in this case, we've got, let's say, a naive system. Antigen comes in to a lymph node, is taken up by dendritic cells, which then process the antigen and stick the peptide onto MHC. And a naive CD4 cell will come in, recognize it, get activated by the dendritic cell, again, signal 1, signal 2, signal 3, and it will then realize that it's supposed to be a helper T cell, and it will move from the paracortex into this kind of interzone between the paracortex and the cortex and wait around, hoping that a B cell that presents that very same peptide is going to come up and find it. So meanwhile, you will have this early, what's called an extrafollicular focus is going to happen, where B cells are getting activated by antigen. So now the protein antigen has come in also and been collected on follicular dendritic cells in the follicle. B cells will come out of the circulation, the same as T cells, naive B cells, and they will pick up that antigen and become stimulated by it a little bit. And when they do, they will then move into that interfollicular zone. And if the antigen that they picked up is the same antigen, and then they will process that antigen and stick on their class 2 MHC molecule, that very same peptide. And so that will allow the T cell B cell interaction to be complete, and the two will recognize they're supposed to be together. And so that now they're going to have a love fest, basically. And so what happens is the T cell will stimulate the B cell through the T cell will stimulate the B cell by upregulating its CD40 ligand that will that I just told you about, and secreting cytokines that will get this B cell going. And meanwhile, the B cell has gotten the T cell going by CD28 interaction and also the MHC, providing MHC for stimulation. So, excuse me. So between those two things, then the B cell will become activated enough to create this what's called a short-lived plasmablasts in this what's called an extrafollicular, because it's outside of the follicle, focus. So you'll get a burst of IgM production by these short-lived plasmablasts, and again, this T cell that was a helper T cell is going to differentiate further into an extrafollicular T helper cell that is going to be turning into a TFH cell, or a follicular T helper cell. So it's going to upregulate basically chemokine receptors that are going to make it move in, and the activated B cells will also now move in with the T cells into the follicle, where they're going to set up and create a germinal center out of that primary follicle. So now this slide is about the germinal center reaction. Okay, so these two guys have decided they love each other and they move in, and in what's called the light zone of the follicle, our follicular dendritic cells still have antigen on them. B cells will grab that antigen and present it to this T follicular helper cell, who will then stimulate it. And so they're both stimulating each other. And the B cells will then move into what's called this dark zone to proliferate. And then they'll move back into this zone, grab some more antigen, and activate some more TFH cells. So this can go on and on until literally you can see with the light microscope, everything gets squished because there's so much proliferation, and you can see a germinal center is formed. During that time, the B cells will somatically mutate their expressed antibody genes, so the rearranged antibody genes. They will also undergo affinity maturation because they'll start competing with each other, and the clones with the best binding B cell receptors will win. And they'll also undergo isotype switching, and they'll also create these plasma cells and memory B cells. The plasma cells will become long-lived producers of antibody. So they'll move either to the bone marrow or, if possibly, the gut, and become long-lived secretors of antibody. All right, and so, and again, this is just explaining how this whole thing happens again. You also, let me remind you that the notes, you should be able to see if you've downloaded the slides, and you can write notes right in those note boxes, and I have notes in there for you as well. Okay, so what do we mean by, some of these responses? So first of all, in differentiating into plasma cells or plasmablasts, we have these, quote, central memory B cells, and also, central memory B cells. So these naive B cells have now differentiated into memory B cells, and I'm calling them central to remind you that what they know how to do is to go to the secondary lymphoid tissues looking for antigen by going to the follicle. So it's not just naive B cells, but also these memory B cells go to the follicle looking for antigen. And those guys have on their surface antibody, and they have, it's just an RNA, alternative RNA splicing, where they have a transmembrane region. And then when that cell becomes a plasma cell, it gives up the transmembrane region. Instead, the splices on this tail piece, which allows the heavy chain to be made as a secreted heavy chain instead of a membrane-bound heavy chain. Class switching requires signaling by CD40 ligand from the TFH cell, and also either cytokine and or LPS and other signals. That's signal 3, this is signal 2, we've talked about before. And depending on what the cytokines are, you're going to have different class switching happening. If it's just plain old LPS, you're just going to have kind of IgM production pretty much. And this is the constant region of the genome here. So we've got our rearranged VDJ, mu, and delta. And see these little S's? Those are switch regions. So a protein called activation-induced cytidine deaminase is what is responsible for class switching and somatic hypermutation. So what will happen is you will get switching, which will cut out this region and bring, for instance, this S switching region, cut out everything in between and jump over to here, where now you're going to be making a gamma-1 heavy chain or an epsilon, so there's IgE heavy chain. And so in different cells, you can have both these happening. But the point is, this is all run by transcription. So these regions get transcriptionally activated, and that's how the aid knows to switch over to these regions, because they've kind of opened up on the chromosome. And similarly, if you have TGF-beta, you can go to these other, making IgA, and in this case, gamma-2b. This is the mouse. The mouse makes IgG1, 2A, 2B, and 2C, depending on if it's a Bald-C mouse or a black 6 mouse. It's either 2A or 2C. And humans have IgG 1 through 4. So it's pretty much the same idea, though. Okay, and then affinity maturation. Well, what does that even mean? Somatic mutation is happening, and most mutations are not helpful or they don't do anything. But some amino acid changes induced by somatic mutation. Again, this is by activation-induced cytidine deaminase, better known as AID. You can get better binding. And so that tiny subset of B cells that have better binding will also have the edge during affinity maturation, because again, the low affinity guys are going to drop out while everybody's competing for antigen in that germinal center. And so this is bringing us back to immune repertoire sequencing. Now, these data were generated from monoclonal antibodies that were made against what we call a hapten. So people a long time ago would take something like ovalbumin protein and then covalently link to it a synthetic small molecule, drug-like molecule. And typically it's like dinitrophenyl is what a lot of people used. And as a result, you could take that DNP and stick it on like bovine serum albumin and find monoclonal antibodies that bind to DNP. And because it's such a tiny thing, the chances were that you'd be using the same immunoglobulin genes, and as a result, it was easy for people to look at lineages because you're going to see the same gene over and over again. And over time, after day 7 from the primary immune response, day 14 from a primary, and then they boost the animal, give it a secondary response and boost it again, get a tertiary response. And you can see in these lineages a buildup of mutations, and some of them are associated with. You can also see that the monoclonal antibodies go from a lower to a higher affinity, which is the inverse of the KD, essentially. So, so as a result, you are seeing affinity maturation, and that's going to be related to clonal lineage. And nowadays, we can just do deep sequencing and look at the whole schmear instead of having to make separate monoclonal antibodies. Another weird thing that I'm going to just briefly point out is sometimes with these big germinal center responses that are going on and on and on, because the antigen is persistent, such as with malaria, people have found that little bits from other genes elsewhere in the genome will get incorporated by AID into the B cell, the recombined expressed antibody genes, which is pretty weird, and it can be seen in various kind of weird ways. So if you're interested in that, you can read about it here. I'm not going to go into it too much, but it looks like it's fairly common when it comes to certain kinds of parasitic infections. Again, a very strange thing that the immune response does. And that, for instance, that little domain I just showed you, that actually binds to malaria bugs. Okay, so now I want to talk to you a little bit about co-stimulation in determining the type of immune response generated, including how peripheral tolerance can be generated. And again, this is all based on the signal 1, signal 2, signal 3 idea. So again, I'm just taking an up-close look here. So T cells, the helper T cells, are getting activated by the B cells through signaling through the T cell receptor. and also through CD28 getting activated by B7 that's going to be upregulated on B cells. Okay, and then the B cell is getting activated, signal 1, by virtue of binding to antigen and CD40 ligand binding to its CD40 and also cytokines activating it. So that's what you see here. And so they both are getting activated to push this germinal center response. Now, what happens if you don't have CD28 co-stimulation for a T cell? So this is a dendritic cell. So this is a naive T cell just starting out, and it's, let's say, CD4, and it's getting antigen. And the answer is that if the dendritic cell is not activated and therefore isn't upregulating B7, then this T cell is just going to kind of become anergic and eventually like give up and die because it's not getting stimulated and kept alive. Whereas if you have B7 CD28 and cytokine stimulation, then this guy is going to want to continue to live, partly because it will it'll start producing IL-2, which is a growth factor for T cells, and that'll keep it alive. So it'll be able to survive and continue in its differentiation pathway. Excuse me. Now, again, there's a way of blocking that T cell activation by, and that is that CD28 can get replaced with CTLA-4. And that's when the T cell is basically saying, okay, I need to shut down this signaling. I'm going way too far on this. And that's going to be similar, but it's not an ITIM mediated response. What basically happens is B7 is just blocked by CTLA-4 such that CD28 can't get to it. So it's more like steric hindrance than ITIM-based suppression. Okay, so, and this is a little therapeutic thing I thought I would throw in here. So if you have uncontrolled inflammatory responses of certain kinds, you can actually make a CTLA-4 fusion. So those outer domains of CTLA-4 fuse to FC, which the IGFC keeps it in circulation, keeps the it makes it last longer in the circulation. IgGs last in the circulation for about 20 days. So this thing will hang around your circulation, bind to B7, block it, and prevent co-stimulation. So that's one way that you can kind of knock back inflammatory responses. Anyway, so if you look then at the whole kind of thing of B cells and T cells getting activated, the B cells are not shown here, but it's pretty much the same idea. So let's say you have an infection, your CD4 and CD8 T cells will get activated. You'll see all this proliferation, and then they will contract down, and you'll be left with kind of a memory cells going on here. So you can see you've already cleared your infection early on. And among those memory cells are going to be dominant clones that remain after the contraction. And so that's going to be true for B cells and memory T cells and memory B cells. And they tend, if they're T cell driven responses, oh, and let me point out that CD4 cells not only help with memory B cell responses, but they also help CD8 with CD8 T cell responses. If you don't have CD4 cells, you don't get good memory from CD8 cells or from B cells. So the point is that these memory cells will be much more focused. So again, if you were doing deep sequencing, here you would see like huge clonality be all over the place. But when you get into the memory population, if you were, pulling out cells with antigen, or with, if, in this case for T cells, with basically MHC class 1 or class 2 that has been, gets a peptide associated with it, and then they make these things called tetramers that they slap a fluorescent tag onto, and you can actually sort out these cells or look at them through the flow cytometry, and you can see that you've contracted these populations, and if you were to sort them out and deep sequence them, you would start seeing clonal dominance, right? Your dominant antigen specific clones. So you'd see clonal expansion at the sequence at the sequence level, and you could actually, if you wanted to, for these CD8, CD4, or B cells, you could clone their antigen receptors. Okay, so now we can go on to chapter 4. I hope that that overview was helpful for you. Again, that was all about getting B cells and T cells activated and what the immune response is going to be doing on a cellular basis. But now we're going to put it all together. Yay! And so we're going to be talking about systemic immune responses as opposed to mucosal immune responses, and a little bit about the roles of tolerance and inflammation. And then I'll give you some examples of vaccination, viral infection, and cancer, and so forth. And then a little bit about engineered immunotherapeutics. And, I mean, there's so much stuff out in the literature. I mean, it's like millions of, it's probably billions at this point of pages of, papers that I am giving you like a totally, cartoonish overview of this stuff. But I'm hoping that it's going to be kind of giving you a kind of a framework that you could go deeper into these responses on your own, or with, with your colleagues. All right. So first of all, I want to get into the idea of homing. Homing is super important, but there's kind of a framework that you can always depend on to be thinking about homing. First of all, as you recall, naive lymphocytes, whether they're B cells or T cells, are primed in the secondary lymphoid tissues. And they're primed to proliferate and differentiate into effector and memory cells based on antigen and innate signaling, essentially, to help it out. And again, a signal 1, signal 2, signal 3, that's what you need to get started. And after released into the circulation, they're going to home to specific tissues. So just remember that that's called priming. If you were talking about allergy or or vaccination, because that that's kind of experimental in a way. So it's priming. So these naive lymphocytes, whether they're a B cell, a T cell, a CD4 or CD8 T cell, they have to get primed. So once they get primed in the secondary lymphoid tissues, and that's by virtue of interaction with, for the either dendritic cells, if you're a T cell, or follicular dendritic cells, or if you're a B cell. So if you're in the secondary lymphoid tissues, although B cells can also get activated outside of the secondary lymphoid tissues, outside of the follicles, in, quote, extrafollicular B cell responses. Anyway, so after release into the circulation from the lymphoid tissues, they will essentially follow the lymphatics back to the right heart, go up into the lungs, come back out of the lungs, and then go out the left heart to out the aorta. And then they're going to be looking to go home wherever they're supposed to home to. And so lymphocyte entry as with lymphocyte entry into the lymph node, where they're drawn in again by chemokines, the effector lymphocytes are going to be drawn to the tissues wherever they're supposed to go by chemokines. So they have things called chemokine receptors, but there's more steps to it than that. And the reason why you have more steps is just like why you have signal 1, signal 2, signal 3, is because your immune system wants to be absolutely sure it's doing the right thing, and it wants to direct the response to some degree. So that's why with homing you have 3 types of interaction. You have selectins that are going to cause lymphocytes to what we call marginate in the capillaries and the post-capillary, so that by the time they hit the post-capillary venule, they're rolling along and they can walk out at the level of the post-capillary venule. Selectins, like their name applies, means that what they bind to is going to be carbohydrate moieties that are attached to proteins typically. Chemokine receptors are the second thing in the response in this orchestration of homing. Chemokines will be secreted from the tissues, and they will actually bind or be available at the capillaries, because the capillaries have little gaps in them, and so these chemokines can kind of creep in. And chemokines are interesting because they like to bind to extracellular matrix proteoglycans and stuff like that, heparin. They like to bind to that kind of stuff, so they actually are sitting there. They're not like a cytokine. They're a small molecule, like a small protein like a cytokine, but they literally can bind and stick to the extracellular matrix, which is great because when that sets up a chemokine gradient that cells can get attracted to, and they can feel the gradient with their little receptors and walk up that gradient into the tissue. So it sets up a chemokine gradient, starting with being in the vasculature. And then finally, there are integrins that allow the rolling cell to actually stop. And what happens is, again, chemokine receptors on the lymphocyte will alter the integrins such that from being a weak binder, it will become a tight binder. So it literally changes the structure of the integrin so that it now binds tightly to the ICAM or whatever it's binding to that it's specific for, that allows it to stop so that the cell can stop and walk up that chemokine gradient into the tissue. And you're going to see this over and over and over again. And I am not going to get into too many details here because it'll make you crazy. So, but the point is, depending on what the combination of selectins, chemokines, and chemokine receptors and integrins and ICAMs, that will dictate where the cell's going to go, essentially. Now, again, that's going to be a primed lymphocyte that has entered the circulation knowing what it's supposed to become because it was told to be that way by what was ever going on in the secondary lymphoid tissue when it went from being a naive cell to being an effector lymphocyte of some kind. Okay, so here's a little picture, and this is selectin. So this is L-selectin on and in this case, this is the same darn thing's happening for neutrophils and monocytes. Neutrophils, so this is actually a neutrophil here, but it's the same exact idea. So there's L-selectin on the cell, and it can bind to a carbohydrate moiety and allow it to marginate and roll on this vasculature. And then, as you know, a lot of neutrophils in a place, well, that equals pus. So, of course, you can get a huge neutrophil influx, but this is how they get out. There's a chemokine, in this case it's CXCL8, that is then sitting on the vasculature around where the capillary is. So this guy's been cruising through an artery, then an arterial, then a capillary, and then at the capillary, post-capillary venule stage, it recognizes this chemokine that has been secreted or made in the tissue and released. So now you've got this nice little gradient. And when the neutrophil sees it, it's rolling along on this selectin, because it's the endothelium of the vasculature has gotten inflamed, and it's upregulating these selectins. So it starts rolling along, and then it sees this chemokine. It's like, oh my God, chemokine. So it starts signaling, and then it makes LFA1 change its, this is an integrin, change its structure to become high affinity for ICAM1. That's also upregulated here because it's an inflammatory situation. And that allows this guy to start walking, to stop and start walking. So it's allowing it to differentiate into something that wants to now do chemotaxics. So it's going to be walking up this gradient into the tissue. Okay, so that's rolling adhesion, tight binding via the integrin because of the chemokine, and then what they call diapedesis, or again, you can call it chemotaxis, and migration into the tissue. So, and in this picture, I did this again just to really try and hammer this idea that this is any leukocyte really that is in the blood, and the chemokines are being made in this situation. It's saying by a macrophage. It could be a bunch of different things. Chemokines are forming a gradient, and that gradient is what these guys are going to walk up into. And the chemokines are sticking to fibrin, fibronectin, extracellular matrix. And again, it's usually proteoglycan of some kind. Okay, so now that you know that part, let me just point out that effector T cells then can home to tissues using special combinations of selections, adhesion molecules, and chemokine receptors. So in this case here, we're talking about T cells that were primed to home to the intestinal epithelium. So here is a capillary in the lamina propria. I think I showed you lamina propria before. Here's the intestinal epithelium, and this is supposed to be like a villus or something here. And so this guy is going to be recognizing MADCAM1 that's upregulated in the vasculature of the intestine. Okay, so this guy, this T cell upregulated this alpha-4 beta-7 integrin, for instance, in the CCR9, knowing that that's where it's supposed to go. And so it gets out doing just that, right? And can then leave the vasculature and get into and be right underneath the epithelium here of the lamina propria, for instance. That's in the gut. In the skin, you can have the same thing happening, the exact same thing happening, but now instead of the, the selectin here, I don't know, it's L-selectin or whatever, but in this case, the selectin is actually E-selectin that binds to this carbohydrate called CLA that's on a particular protein. And anyway, so this thing is getting out with a different chemokine receptor, CCR4, because it's been primed in the primary lymphoid tissue a long time ago to recognize CCR10 and CCL17. So it's checking out CCL17, and then it will actually, in this case, go even further because it's got CCR4 to recognize CCL17, and then once it gets into the tissue, it can go even further and recognize CCL27 through the CCR10. So the point is that maybe this is a smaller gradient that's only coming out this far, whereas the CCL17, the gradient is all the way into the vasculature, if that makes sense. So you can actually have layers of this stuff, just so long as the T cell has, or B cell has upregulated the proper chemokine receptors. Okay, so I'm going to now take a 5-minute break. We're halfway through the slides, yay, because we're supposed to be done in another hour and 10 minutes. And so I'm hoping that I can get through these other slides with you, and then have time for a few questions. So we'll be back in. Well, I'm going to sit here, but I'll be back. You guys can take a little bathroom break or get a cup of coffee or whatever, and just be back in 5 minutes. I'm going to start up again in 5 minutes. Thank you. All right, then. So I hope everybody's back, and I think that was 5 minutes. Probably isn't exact. So now what I want to do is put together the cutaneous, the kind of the overview of the cutaneous immune response. And cutaneous means skin. So this is the immune response in your skin. Duh. Okay, so we first have to think about what the skin looks like. So your skin has 2 layers to it. The epidermis. Then there's a, for pretty much all epidermal layers, well, and it's also true for endoderm, endothelial cells. So for the epidermis, there's multiple layers because we need a good, strong barrier to hold in moisture and all kinds of things. There's a basement membrane, and just above it is a stratum basale, which is that single layer of cells here. Those are the stem cells that your skin gets renewed off of all the time. So they get renewed and turn into keratinocytes and flatten out and do all kinds of stuff to then form the barrier of your skin. And there are certain cells that are in that epidermal layer, and one of them that's really interesting is the Langerhans cells. There are other dendritic cells. It's a type of dendritic cell, and there are other types of dendritic cells in your epidermis looking around for antigen, basically. And then also, you'll have mast cells. So if you've ever been bitten by a mosquito or something, those mast cells are what are making it really itchy by producing histamine and stuff. So they can get activated a bunch of different ways. And so these dendritic cells, for instance, can pick up antigen. Let's say you've got a little infection, you got a scratch or something. The dendritic cells or Langerhans cells can get activated and then move into the dermis, this lower section here, where there's a good blood supply, and travel up the lymphatic vessels, just like as usual, to a draining lymph node where they will present antigen and prime lymphocytes to recognize that antigen and to prime them to home back to the skin. So they will then, those lymphocytes in the regional lymph node will get primed, and they will come back through they'll get primed in the lymph node, go into the efferent lymphatics to the right heart, go through the right heart up to the lungs, back out of the lungs, out the aorta, and eventually get to a vessel where if the chemokines and other integrins and ICAMs and all that are right for the cell, it can then, that lymphocyte can then enter the dermis. Now, for the most part, T cells won't enter, but B cells, because this is a systemic response. So B cells will get activated, but they will go somewhere else and secrete antibody, typically to the bone marrow for long-lasting antibody responses, or the spleen, for instance. So it tends to be B cells and T cells here. And also in the epithelium, you will have some of these very innate-like gamma delta T cells and other innate-like cells, ILCs, in the epidermis there, kind of telling the Langerhans cells and other cells what they ought to become when they're going to go to the regional lymph node and prime. So here's I think a little bit better of an example. So again, there's antigen, you've got dendritic cells and Langerhans cells picking up that antigen, getting activated, being affected by a whole bunch of other types of cells in the epidermis, and leaving, going to a draining lymph node, activating a naive T cell that's then going to upregulate all the things it needs to then home to the efferent lymphatic, the systemic circulation, back to that kind of tissue, leave through a postcapillary venule, and either go to the dermis or even into the epidermis. And I'm not going to get into all the chemokines because I think it'll drive me and you crazy. Okay, so what about mucosal immune responses? What about mucosal immune responses? Oh, and let me just go back for 1 sec. So this is kind of what's happening when you get vaccinated. When you get vaccinated. Again, you know, it's not B cells going into your skin and secreting antibody. Why would they do that? You need the antibodies systemically, so in your tissue, throughout your tissues, to protect you. So the T cells will come back, but the B cells will be going to again to the bone marrow or to the spleen, for instance, to secrete antibody. And if you got a great vaccine, then it'll be a long-lived B cell plasma cells, long-lived plasma cells that seek a special little niche in the bone marrow to live for years. They can live like your entire lifetime. A good example is the attenuated measles, measles virus vaccine. That gives you lifelong immunity, typically. Okay. Anyway, okay, so now I need to tell you about so you're going to get a whole bunch of anatomy from me at this point, because again, I have to explain what the mucosal tissues are to you in a lot more detail. So basically, the mucosa are epithelial surfaces that are wet because you've got mucus on them. And so you can think of it that way. And they are also in a lot of cases, the outsides of your insides. So for instance, your lacrimal gland is epithelial, you know, your tears come out of it. Your salivary gland, saliva comes out of it. The whole inside of your mouth is lined with epithelium, right? And it's mucosal epithelium. So we'll get into what that looks like in a minute, but same thing goes for all the way down to your alveoli in your lungs. The biliary tree and the pancreatic ducts coming out from your duodenum and going up into your liver and so forth, or from the urethra to the bladder, to the kidneys. That's all going to be epithelial cells. And then also the scarily, the lining of the fallopian tubes, which then is opens up into the peritoneal cavity. So, yikes. Okay, so that's where all of your mucosa are. And I want to talk a little bit about the gut, especially because the gut is typically about 80% of all of your lymphocytes are associated with the gut associated lymphoid tissue. So, all these tissues, the liver and so forth, are all the arteries going to them are just like normal, but you can see here that there's this venous drainage from the esophagus all the way down to the colon that is draining these lymphoid, your GI system, and that's taking everything to the liver, essentially. And that is because you are digesting food, right? And you're absorbing it. They're not shown here as the small intestine, which happens before the large intestine. Anyway, you're absorbing food and water and moving salts around and all kinds of stuff. And so there's protein being absorbed, all that. And so that's anything that's being absorbed and going into the venous system is going to go through the liver to get cleaned up and looked at. Similarly, you're going to have kind of a system that I'll show in the next slide. Oh, this is kind of a bad slide. I'm sorry, you guys. Of the lymphatic system. And the lymphatic system is covering, they're just showing the, well, here, let me start with this. This is showing the small intestines, and you can see that you have all of these lymph nodes draining, this miles and miles of small intestine that you have. And that is going to then dump into this thing called the chyla cisterna, which is a little vessel that goes way up like that and dumps into the thoracic duct. So this is like major lymphoid system going to the thoracic duct. And the large intestine also has a bunch of draining lymph nodes as well. So I just want you to appreciate the very cool lymphatic drainage that's happening in different parts of the, especially the intestinal system. So what's happening here then, because at the same time that you are trying to protect your gut, you're also absorbing food. Weird. And so I'm just letting you know that in general, so here's the intestinal lumen. It's full of bacteria. And as you go south, from the stomach to the large intestines, the amount of bacteria slowly but surely increases. So your stomach, for instance, is sterile. But it's in your, mainly in your small intestines, in the jejunum, where you've got really big villi, and you're absorbing amino acids and sugars. Those are going into the venous circulation to go to the liver through the portal vein. And then fats and all that are absorbed through the afferent lymphatics and going up through the cisterna chyli and all that to the thoracic duct. So in case you're interested. And but the point is, you want to keep out these intestinal bugs. And so you've got this nice, the blue here is supposed to be a layer of mucus that is kind of preventing these guys from invading this nice epithelial barrier here, okay? So now what happens though is that when a T cell or a B cell gets activated by antigen, it's going to do what it always does, climb into a draining lymphatic vessel and find the nearest lymph nodes. You saw those things, those mesenteric lymph nodes, and that's where you're going to be priming naive B cells and T cells, which are then going to leave via the efferent lymphatics, get themselves in the thoracic duct, go through the heart, come back out, and home back to the tissue. Okay? Now, this is kind of gross because it's brown, but this is to let you know that in the small intestine coming out of the stomach, so you've got the duodenum, then you've got the jejunum, then you have the ileum, and it's getting browner and browner because you're getting a higher and higher colony-forming units per gram of stuff in there. So it starts out mostly food and ends up mostly bacteria, right, in the colon. And so this is why there's so many bacteria in the colon. This is part of the reason why in here you want to be kind of careful, because you've got a lot of bacteria in there and you really don't want them to invade your tissues. Here it's a lower bacterial load, and this is the place where Peyer's patches are. They're at the end of the ileum before you get into the large intestine. You'll see these things called Peyer's patches, and if you were to scope somebody, you would see all these little bumps because they're kind of big. They're kind of like tonsils almost, tonsil bumps, if you know, essentially. And so I think what they're doing is kind of protecting this system from getting a lot of leakage of all these guys into your small intestine, because what you want to be doing here is not have it sterile, but at least sterile enough that you can be absorbing your food. And as a result, you have to have a lot of tolerance on this end here. You want to create tolerance. You don't want to have an inflammatory situation. So you have a lot of what are called Treg cells or CD4 cells that actually suppress immune responses. And you need them here and here to make sure that you don't have an inflammatory bowel disease. And actually, most of the inflammatory bowel diseases that you hear of are regarding the colon. Okay, so again, I'm just going to walk you through this. I hope I'm not a broken record, but it's kind of complicated. Is here we have the intestinal lumen, and then you have your mucosal epithelium. Okay, and then you have this thing called the lamina propria, and then it's better to show it here. And then underlying that, you've got like 3 kinds of musculature. And then at the other end of this, you've got, you know, it's a tube, right? You've got the inner lining to the tube, you know, it's fibrous connective tissue and stuff like that. So if this is the tube of your gut, right? Okay, so the mucosal epithelium plus the lamina propria, and there should be a basement membrane here. Oh, sorry. The basement membrane is here. I'm being stupid. But the lamina propria plus the mucosal epithelium, that constitutes the mucosa. Just those 2 things. Okay, so now you've got certain kind of cells, they tend to be CD8 cells, that can be located in the epithelium, intraepithelial lymphocytes. And you've got pretty much your dendritic cells, which weirdly enough, but it's really true, they can reach out and touch these little guys, these bacteria and stuff that are in your gut, or food antigens or whatever. And then they can actually enter, but they're in the lamina propria, as are B cells and other T cells. And then, of course, your Peyer's patches in the ileum, which are like very highly structured. They've got B cell zones and follicles and T cell zones. So they're like a little lymph node almost. Anyway, the point is that dendritic cells can get activated in the tissues or in the Peyer's patch, and go to a mesenteric lymph node or set up a little immune response right in that Peyer's patch. And cells can then go to the heart, naive cells can get activated primed, and then leave, go to the heart, and come back through the vasculature again, that through the blood vessels that are running through the lamina propria, or that are entering the Peyer's patch. And again, the Peyer's patch are specific little structures that are mainly in the ileum. Now, the plasma cells in the gut will home to what they call the crypts. So you see a villus here, and then it kind of an in-pouching. Here's some crypts here. And what you'll do is you'll get plasma cells will go down to this bottom of the crypts, and they will secrete IgA, and they have specialized epithelial cells there that can move that IgA and IgM into the mucus layer, which then those antibodies will pick up, bacteria and stuff like that, and keep it away from the epithelium, keep them from invading. And goblet cells are the epithelial cells that secrete the mucus. And down here at the bottom of the crypts are the stem cells for the villi. And what happens is the stem cells start to differentiate. They come out of the crypts, and sometimes they become a mucus secreting cell or some other kind of cell. And then what happens is the epithelium sloughs off, just like your skin sloughs off. The epithelium sloughs off the tip of the villus. So the structure doesn't slough, just the epithelial cells slough. And that's good, because you want to renew them all the time, because they're getting beat up and all kinds of things, from being exposed to this environment. They're also little isolated lymphoid follicles, I should say. So I hope that makes sense to everybody. And this is again, the small intestine. You can tell because the big villi, the villi are shortened in the colon. And you can see this gigantic Peyer's patch. So where are we? We're in the ileum. We're just about ready to go into the large intestine. And this is just if somebody wants to, I don't really want to go through all this, but the point is vitamin A can help to get dendritic cells to understand that they're supposed to be priming T cells or B cells to come back to the epithelium through these chemokine receptors. The thing that is very cool about the gut, I think, is that you can start up a gut homing response, and depending on what the chemokines are, you can get broad homing to, like, it starts in the gut, but you can get homing to the gut, the lung, the genitourinary tract, and the lactating breast. So the point is, for instance, whatever's going on in mom's intestine and what kind of bacteria she's trying to hold off, the baby is going to drink those IgAs, because remember they're secreted into the lactating breast, just like those antibodies are secreted into the gut lumen, and because it's the same clones, and they're going to protect that baby from those antibodies. And when the baby is born, it is picking up mom's bacteria to begin with. So kind of a cool system. Okay, so what about IgA class switching and so forth? So the class switching is going to be IgA, and that's because TGF-beta is pushing B cells to class switch to the alpha, to use the alpha chain. In people, there are two different alpha chains, by the way, and I'll mention that in a minute. And I gave you guys, in case you're interested, a little thing that you can read on it down here. Anyway, so you've got luminal antigen, and just to give you some more stuff, if it's a Peyer's patch, which again, this is almost like, a tonsil or something, it's pretty well structured. On top of it is this thing called an M cell. So this is a modified epithelial cell, an M cell, that is literally made to grab antigen and feed it to dendritic cells and directly to B cells, so through the follicle, not directly to B cells, to follicular dendritic cells in the follicle, so that you can have B cells and T cells find each other, fall in love, and then get into the circulation and come back out. And the IgA secreting plasma cells are going to park themselves at the base of the crypts in a whole lot of different places, right? So they're getting activated here, but they're going to park themselves in a whole lot of places, depending on what the chemokine receptors are that they were primed against. Those are T-dependent follicular, FO is follicular, B cell responses. You can also get T-independent extrafollicular responses. They tend to be with more innate type B cells. And that's just happening through dendritic cells, again, getting activated, secreting TGF-beta, and these cells that are more likely to just kick off a plasma cell response, maybe a short-lived one, not always. Anyway, these guys tend to have not a whole lot of somatic mutations. You'll get IgA and IgM. And IgAs and IgMs are both secreted into the gut. And I'll explain why in a minute. It has to do with the fact that they both have J chains on them. Anyway, so TGF-beta also is something that tells T cells to become T regulatory cells and to suppress responses. Because remember I told you before, the going thing, unless you have invasion of this nice barrier into, by bacteria or viruses or something, you're going to have a pretty nice situation. So long as the mucosa is, intact, you don't want to cause any problems. So the T cells are actually there to suppress. They recognize antigen, but they are suppressing immune responses by virtue of kicking out TGF-beta and telling the T cells dendritic cells are kicking out TGF-beta, which is telling the dendritic cells to prime the T cells to become T regulatory cells, which then will actively suppress immune responses by secreting IL-10 and doing a bunch of other stuff. Okay, why am I telling you all this? I've got to remember this. Oh yeah. Okay, so again, as I told you before, IgA secreting plasma cells tend to live in the crypts, and this is just a better picture of crypts. These are villi, and in case you want to get confused, microvilli are on top of these epithelial cells, even further increasing the absorptive tissue in especially the small intestine. So you can see here in the small intestine, I really like it because it shows big villi, and you can see in the large intestine, you've got limited guys. And I can tell you that the homing signals to get to the small intestine are different from the homing signals to get in the large intestine. Anyway, so what you will get then at the base of these crypts are plasma cells that will then secrete antibody that's then going to go into the mucus layer and follow its way out the poop chute, essentially. Okay. And how's that done? Well, in the crypts, you will get an IgA or an IgM, if it's super innate, producing cells. And IgA, if it's dimeric, is held together by a thing called a J chain. That J chain allows it to get picked up by this thing called the poly-Ig receptor that's on the basal side of the mucosal epithelial cell at the base of the crypts. So it will then grab this thing. It's a receptor. It will grab this thing by the J chain, and as it transcytoses to the luminal side of the epithelial cell, it literally chops itself off, and now this receptor component, which is called the secretory component, remains on the dimeric IgA, and it will remain on the pentameric IgM, and that allows it to bind to the mucus layer and stay right here, you know, right nearby, but moving along and grabbing bacteria and protecting the epithelial layer from bacteria that might want to go further and invade. Okay, so so those are no, that's how the mucosa works, kind of. I hope that was clear. Anyway, okay, so I want to now just get through a few examples of immune responses, and I'm going to kind of whip through these kind of quickly because I don't want to like say this stuff to you like 8 million times and drive you up a wall. Okay, so first of all, let's think about vaccination, because vaccination is the way that we've learned a lot of stuff about the immune response. So the first vaccination is called the primary vaccination or the priming vaccination typically. And then later vaccinations are called secondary or booster vaccinations. Okay, so when you prime, let's say you're immunizing a rabbit or somebody with something. And so you prime the animal with the vaccination, and you will have a lag time. And then you will have an immune response. It'll mainly be IgM, and it'll kind of flip about right around in here to an IgG response, and then it'll go down after a while because your plasma cells will poop out. Okay, so that would be your primary response. But just remember that you have made a bunch of memory B cells, and if this is a protein antigen, you also have follicular T follicular helper cells, and now they're memory cells too. And memory cells respond a lot faster and a lot stronger. Why? Because you've had clonal expansion. So if you were to do deep sequencing, you would see clonal expansion, and if you used antigen to sort out your cells, you may see some dominant clones even early on. But after your secondary response, once you boost again with antigen A, you'll get a much faster, the lag time is shorter, and you'll get a much stronger response, and it will already be pretty much completely, if it's systemic, IgG. But you know, the same thing holds for if I don't know, if you had an oral vaccine, for instance, and this was happening in your gut. So so beautiful. And if you've got really good adjuvants, then those plasma cells are going to go to the bone marrow and become long-lived B cells. So let's just talk about COVID for a minute. So the COVID vaccines are two different kinds right now of gene delivery vaccines. And what they are doing is, the one is basically the RNA vaccines are RNA molecules that are inside a lipid bag, and they call it a nanoparticle because they're about a good size for a dendritic cell to want to take it up. They're shooting that puppy into your arm. And dendritic cells are taking it up, getting activated. Why? Because RNA is in a place that it ain't supposed to be in those cells, and that makes the whole thing, the cell, think it's infected, and as a result, it upregulates all kinds of innate stuff and immediately goes for the nearest draining lymph node. And we know it works pretty well because you feel horrible when you when you have the vaccine for a couple of days, because it makes you feel like you've got the flu. Why? Because you're getting an interferon response, which is exactly what you want, and why old people like me, we have great responses to these vaccines. Whereas if somebody pops me, for instance, with a protein-based, not very good adjuvant vaccine like your typical annual flu vaccine, you know, they got to give me an extra dose because I'm old and I won't really have a very good response. So these are great freaking vaccines. Now the adenovirus vaccine, so that's the J&J. Why is it only one dose? Well, because it's an adenovirus. You make a response to the adenovirus itself, and as a result, when they go to boost people a month later, they probably should wait longer. You've got plenty of antibodies that clears the thing out before you can create a good secondary response. I think that's what happens anyway. And the AstraZeneca vaccine, well, that's a chimpanzee adenovirus. It's not a human one. And so that works a little bit better, as does there's a Russian one too that I think is probably pretty good, that is 2 different human adenoviruses, one for the prime and one for the boost. But, you know, those adenoviruses are a little scary because we don't know. We do know that some people make antiplatelet antibodies, so they're having an autoimmune antibody response. And I haven't seen any data yet to show if they're making other antibodies as well. Like, nobody talks about that. It's only 1 in a million or something, but boy, if you've got alternatives, why even bother with 1 in a million, to my mind. Anyway, so those vaccines, and then Novavax just came out with a protein-based vaccine, and they've got a really good adjuvant. It makes you feel horrible, maybe not as horrible, and they're getting great efficacy from it. Another vaccine that has that's protein-based, that has great efficacy, is the Shingrix vaccine. And again, it's 100% because of or mainly because of the adjuvant in there. They've got a really good adjuvant, and even old people like me, that's a vaccine for people over 50 to prevent shingles, and it is one hell of a good vaccine. It's 90% efficacy in old people. Okay, I'll quit talking about that. So now let's talk about viral infection. So viral infection, we're going to break this down into mounting an immune response and removing the vaccine. Then what do you have left? And so it's kind of like how vaccines work too, but a little, yeah. So, so you start off with an infection, and here's your viral titer going up. Your immune response kicks in, and it starts going down. So the point is the innate immune response is super important. So what you want, and this is exactly what the RNA vaccine and the adenovirus vaccine do. They make your cells think that there's an immune response, a viral infection, and as a result, those vaccines or an actual viral infection will cause a type 1 interferon response. And type 1 interferons are made by different kinds of innate immune cells and a lot of different kinds of cells, including epithelial cells. And what it does is that it's like an alarm. It alerts all these cells to upregulate antiviral protective mechanisms in the cell, including upregulating, for instance, class 1 MHC and all the stuff that processes antigens, because there's a lot of viruses out there that have mechanisms for undermining class 1 MHC, because they know that's how they get detected and cleared out by CTL. And so they really try to evolve ways of getting around class 1 MHC. Anyway, so you've got a type 1 interferon response that is going to help to prime dendritic cells to push a CTL response that's going to be antigen specific against that virus. NK cells are kind of stupid, but they're really good. They're kind of like a CTL. They kill cells in the exact same way. And for instance, if you have a virus that's undermining class 1 MHC, NK cells will go, hey, where's the class 1 MHC? I'm going to kill that puppy. And they will kill that cell, which is kind of cool. Anyway, and NK cells also, if you make antibodies, can kill cells through ADCC. Meanwhile, the CTL response is coming up. You're clearing your virus, so now you can have a contraction. And why do you want contraction? Because the niche. You don't want to fill up your CTL or your T cell niche in your body with these guys that you really don't need anymore. So it contracts, but you're now going to be left with good memory, and some those are going to be called central memory cells. Why? Because they go and look at your lymph nodes. They go to the secondary lymphoid tissues looking for more antigen. And meanwhile, you're going to have an antibody response. And notice the antibody response is really just kind of helping to clean things up at the end. But again, if you have, like, let's say a COVID infection, or you have the vaccine and wait longer than a month, I would say, like out maybe this far, this is days, so this would be like 6 months later, you will see that with COVID infection, if you catch people 6 months later rather than after the first month or two after initial symptoms, you will see a ton of, maybe there won't be as many B cells, but they will be much more loaded with somatic mutations. Their antibodies will be higher, tighter. They will have undergone affinity maturation, is the point, whereas they won't have early on. So that's pretty interesting, and that's what we want vaccines to do. So it's my idea, and a lot of other people's ideas, that probably getting you out to wait 4 months, get your initial prime, and then boost, and maybe they'll have to boost people a year later, I don't know. But after that, you're probably going to have a hell of a good immune response against the COVID vaccines, which also, by the way, have good coverage for all of the known variants that are scary. Okay, so you have innate immune response happening, and type 1 interferon, NK cells, a cellular and a humoral, I guess you could say, response. And then you have your B cells making antibodies that will neutralize. The antibodies will also bind to infected cells that have viral components on their surface, and NK cells can kill them through ADCC because NK cells have Fc gamma R1 receptors on their surface. And then also, of course, you're going to make CD8 cells that are going to kill infected cells that are showing class 1 MHC on their surface. Okay, immunity to viral infections, right. So this is just a recap of all that. And let me just add that if you have a persistent virus like HIV, then after a while, you're going to start seeing exhausted B and T cells, and that's due to things like upregulation of PD-1 and so forth. And again, part of that is that their cells are getting exhausted and eventually going away, maybe, to preserve the niche, because you want your active memory cells and your naive cells to have space in the niche, your T cell and B cell niches. Okay, and then as I mentioned, immune evasion. Okay, what about tumor antigens? Okay, so let's talk cancer now. So I've got a bunch of stuff on cancer for you guys. I figure you probably like that. Okay, so the tumor may have neoantigens, or it might be upregulating proteins that you normally don't see. And so again, you can have dendritic cells getting activated. And you can see, like, around tumors, you'll see a lot of lymphocytes typically surrounding a tumor. And of course, what you want is those lymphocytes to go in there and kill the frickin' tumor. And so people have been working hard at getting that to happen. So probably in all people with tumors, there are dendritic cells that are picking up mutated antigens, better known as neoantigens. Why am I saying that? Because tumors, they get to a certain point of development. First, they're just growing really fast and their genome looks fine. But once they get to a certain point of disruption of the cell cycle and everything, the cells are dividing faster than errors can be corrected in the genome. And as a result, you get all these clonal lineages with all these different mutations in them. And that's typically when you start seeing the tumor, especially metastases. They're just completely out of control. So as a result, you've got neoantigens, and dendritic cells can recognize those, go up the afferent lymphatic, present to T cells, naive T cells, and then those CD8 cells or whatever, CD4 cells and CD8 cells, can return to the tumor and kill. And that's what we'd like. Now, I want to just point out that dendritic cells, somebody asked me about cross-presentation. Dendritic cells, and in fact, all APC, so that's dendritic cells when it comes to priming, and then B cells and macrophages when it comes to effector responses, they all can present antigen on class 1 and class 2 MHC. All your other cells don't have any class 2 MHC except for in weird situations, and they only have class 1 MHC. So the point is that dendritic cells can pick up antigen from the outside, and that's what this is. Pathways of antigen processing and presentation. So normally, all cell types can take cytosolic proteins. So those are proteins inside the cell, and basically digest them in these proteasomes, and then the peptides get pumped into the ER, where there is class 1 MHC, which cannot fold properly until the anchor residues of a peptide. It's not an epitope, they're called anchor residues, but the correct anchor residues with the correct spacing allow it to bind very tightly to that MHC molecule, which then allows it to leave and to go to the surface of any kind of cell. For APCs only, dendritic cells, B cells, and macrophages, those are going to either be cell surface proteins getting endocytosed, that could be an antibody grabbing antigen and going inside, or it could actually be just like a macrophage just doing its little phagocytosis thing, or even pinocytosis by a dendritic cell. And what then happens is it's a different pathway of processing. It's a lysosomal endosomal pathway, but it's the same thing. Class 2 MHC has to have peptide in there that's allowing it to fold and be happy, and then it'll go up on the surface of the cell. So cross-presentation is only by an APC. It is taking, cross-presentation means taking antigen from the outside. It could be, for instance, let's say you've got an infected cell and a dendritic cell has picked up, eaten some stuff, or it could be just a virus that it's eaten from the outside. So it's picking stuff from the outside or the cell surface, and then it's beginning to process it in class 2 MHC presentation, but some of that antigen, it magically pushes into the cytosolic stream and allows it to get processed and put on class 1 MHC as well. So that's why you can have outside of the cell antigens presented on class 1 MHC on any kind of APC, whether it's a dendritic cell that's going to be priming B cells and T cells. You can see why that's important. And also if it's B cells and macrophages working with CD4 cells. Okay, so activated Th1 cells then can direct cytokine stimulation and differentiation. And there's two ways that they can do that. So this is just the other thing. T helper cells are really good at helping APCs get stimulated. So if you have a T helper cell and it knows that the antigen is, an APC has it, even a dendritic cell. So let's say you've got a memory CD4 helper cell, and it sees that an APC is putting, it's class 2 MHC, the same peptide that it normally recognizes, then it can actually get activated further and dump cytokines on this whole schmear, which allows CD8 T cells then to differentiate even more and to become good memory CD8 cells. You need CD4 cells in order to make good memory CD8 cells. And this is one way of doing it. And the other way of doing it is activating the APC itself. So either the T cell can get activated and dump cytokines that directly on the CD8 cell, or it can upregulate and further activate the dendritic cell to be even more co-stimulatory and therefore directly get the CD8 cell going. So that's solving that little problem there of how do you get, why is a CD4 cell necessary for memory CD8 responses? Okay, so here's some tumor therapies checkpoint blockade, so that's PDF1, as you guys probably know. PD1. Vaccination with tumor antigens could be neoantigens or tumor-associated antigens, but you're starting to ask for trouble there. Adoptive therapy with anti-tumor T cells. Those could be CAR T cells. They could be autologous T cells, so from the same person transformed with T cell receptors that are against the tumor. And also activated dendritic cells, where you feed and activate dendritic cells to recognize a tumor antigen and present it in the to prime or boost T cell responses. And then there's also passive immunotherapy with therapeutic antibodies. So I think that I'm going to end in a couple of minutes because, well, actually, I think I'm going to end here. But these slides are just really telling you how those things happen. But I think you can just read the slides. It's pretty simple. About those mechanisms. The last thing I just want to say in closing, before I take questions, is that the importance of what we call big immunological data to aiding our understanding of immune responses. So I hope I've convinced you with this this set of talks that AIR sequencing, so that's adaptive immune receptor, B cell and T cell receptor repertoire sequencing, sequence data can help look at clonal responses and also provide you a way of, for instance, getting at T cell receptors and B cell receptors that you may want to clone for one reason or another, and to make therapeutic antibodies or to transform autologous T cells to go after tumor antigens, for instance. And also now that we have 10x genomics, of course, getting light and heavy chain or alpha beta chain pairing is something that we can do pretty easily and connect it to phenotype. And not only phenotype, meaning what are the cell surface markers on a cell, but with single cell RNA-seq, you can actually get a pretty good idea of what that cell is up to in a more specific way, which is awesome, because then you can start looking at what cell types are involved in disease processes, what cell types are involved in protection, and so forth. Now, systems immunology, maybe all of you know what this is already, but systems immunology takes big data up a further step because systems immunology involves what they call multi-dimensional data. So that could be microbiome data combined with cytoph data, combined with metabolomic data, and so forth. And so these are completely different kinds of big data, including AIR-seq data, to try to figure out what the main components are in an immune response. So they kind of do through artificial intelligence, neural network analyses, and so forth. They can take things even further in our understanding. But I just want to point out that these things can turn into fishing expeditions if you don't know what you're really going after. So I just want to point out that you'll read these papers and go, wow, that is one hell of a fishing expedition those guys went on, because they're like, well, we're just going to do all this stuff and see what happens. But in fact, the more that you can place this stuff into an immunological or a biological context before you get started, the more likely you're going to get outcomes that you understand and can draw relevant conclusions from. Let me just say. So with that, I'm going to end, and that way we've got about 10, 12 minutes for any questions. Oh, so Jan, are you there?
Hello.
Okay. Yes,
Oh,
we
there
have
we go.
rejoined. Thank you very much. Very informative, illuminating. Learned lots of interesting terms, one of which I probably will not repeat.
You mean poop shoot?
Technical term.
Oh God.
Sylvia, you want to ask your question there?
Yes, so
Added a
I'll
question.
start with you said that the lymphocytes know where to go based on their differentiations. What information do the cell get during their differentiation that tells them where they are needed? For example, if they only encounter the antigen in the follicle or lymph node, how do they know it's originated at the skin or in the intestine?
Okay, well, first of all, the lymph node itself is draining the skin, so it's going to already have all kinds of cytokines and other things that are kind of, I guess you could say, what's a good word? Kind of marinating that tissue already. And just remember that the afferent lymphatics are draining that tissue, so you're getting innate cells that are in the tissue itself dumping out cytokines that are feeding into and altering the milieu of the and allowing therefore cells when they get primed, T cells and B cells when they get primed, to know that they should be picking particular, upregulating particular chemokine and integrins, chemokine receptors and integrins, where that will make them home back to that tissue. So that's what I was kind of barking at you about over and over again in those examples of the gut and the skin. So again, just think about the fact that the actual tissue that you're going to home to through the draining lymphatics, right? Those lymphatic vessels, because the fluid is going up so you're getting a flow of stuff of water up that little vessel, because the heart is pulling it. And if you don't understand that that's what's happening, it's impossible to understand this stuff. So that's why I'm saying just think about the tissue is being bathed, and all of that bath content is not going into the vein, the venule. It's going into the lymphatics, like I told you in part 1. And therefore, all of those cytokines and everything else made by innate lymphocytes and other cells in the tissue itself, especially when they get freaked out, like, if an infection starts, suddenly you've got the type 1 interferons, that's all going up into the draining lymph node. So that is going to then be telling the draining lymph node, here's what you normally do. And if there's an infection or inflammatory response, and now we've got all this other stuff coming into the lymph node, telling us that we've got to freak out and start doing an inflammatory response. So, that's why lymph nodes are so cool, and why they're used for priming, because they're being talked to by the tissue via the lymphatics. I hope that's clear.
Yes, perfect.
Yeah, beating a dead horse, maybe.
I actually have a quick question, because towards the end you were talking about cancer vaccines, and eons ago, in a different life, when I was writing papers about various trends in what the biopharmaceutical industry was developing, one of the things that I looked at relatively briefly was cancer vaccines. But this was, I would say, 10, maybe even 15 years ago, not to date myself. But I'm not aware of a huge amount of progress in the field, nothing like the progress that we've seen in antibody therapeutics over the past 15 years. So I take this to mean, and I'm sure it is, It's a complicated process. It's a difficult process, but could you speak just to a couple of the main difficulties and challenges associated with developing cancer vaccines?
Okay, I'll just go back a few slides. Okay, so I'm just trying to find my slide on neoantigens. Yeah, okay, so here's a problem, is that in a tumor, you don't know what the antigens are. Now, they can use tumor-associated antigens, but the problem with tumor-associated antigens is it's really hard to focus on one tumor-associated antigen, and those are not neoantigens. Tumor-associated antigens are antigens that are upregulated. They're not mutated, and they shouldn't be there, and you hardly ever express them except for maybe when you're a baby or something. And so they look like a new antigen, but they're not. And the problem with that is typically there's some other normal tissue that if you were to incite a gigantic response against it is going to get wiped out by it too, which is kind of scary. Now, neoantigens are those mutant guys, and you have to get the stupid tumor and you have to sequence its genome to find all the mutations. Then you have to analyze it according to your class 1 MHC to see which of the mutations are creating anchor residues that now allow the peptide to bind at your class 1 MHC. So it's highly personalized. And then those peptides could be used. That's a lot of work. It's really expensive. So you see what I'm saying? So that's a problem. And let me just point out, and that's the same thing with these dendritic cell-based vaccines, you're doing the same thing. It could either be a neoantigen or it could be a tumor-associated antigen, but you're asking for trouble. And then finally, I guess I would say with these chimeric antigen receptors, and this is why the Antibody Society loves these things, they're actually single-chain antibodies that are against a tumor antigen. But again, if it's against a tumor antigen, it's got to be on the surface, otherwise it won't work. And it's not a neoantigen, and therefore you've got to be really tricky. Like now they'll have like 2 CAR T-cell receptors, one doing tumor antigen number 1, one doing tumor antigen number 2, so it's more specific for the tumor, that kind of thing. So they're actually getting some traction off of this. But again, it's really hard because now what you're doing is taking cells from the person, so you don't have a transplantation response, and you're teaching them by virtue of just sticking the receptor on there that is then going to upregulate and push the CD8 T cell, so that you'd be adding this, you'd be expressing all of this off a gene in a CD8 T cell. You'd be taking a naive CD8 T cell or maybe even a memory CD8 T cell because they're more likely to have everything upregulate to kill. And what you would do then, it's going to signal, right? So the minute this binds, you're going to push the CD8 signaling CD8 T cell receptor signaling apparatus. It's just that out here it ain't a T cell receptor. So those are some things, and maybe that answers your question a little bit. It's just a whole lot more work, but I think people are getting there, and maybe they'll automate all this stuff eventually.
Well, thank you very much. Actually, I don't think we have any more questions from the audience. Sylvia, do you have any questions?
Yes. So this is, I'm involved in this personally for my work. Some people try to investigate the B-cell immune response in cancer in order to do like antigen discovery, to find new antibodies for cancer immunotherapy. This is what I'm trying to do. But the problem is that sometimes, most of the times, the antibodies that you derive and you clone and you check their activity that you derive from tumors are not really binding like extracellular antigen, are binding like, I don't know, intracellular antigen, so they are not really then suitable to be used to develop antibody for cancer immunotherapy. What do you think about this? So trying to use the B cell in the tumor to discover new antibodies.
Well, I know that Atreca is doing that kind of thing, and I know they've gotten some good results a few years ago. They haven't published again on it. It's not really my area, so you probably know it way better than me. But again, I imagine, like you're saying, there's different. If it's intracellular antigens, let's say, because cells are getting blown up and those antigens are being exposed. There may be, for instance, nanobody technologies. There are a lot of people trying to do intracellular therapeutics using an antibody, but that somehow or other can get moved inside the cell.
Yeah.
I don't know if that's. I don't know how far ahead you and Jan would know more about that than me. And then the other thing is, well, I guess that what you could do is do an informatic approach where you would, for instance, may be able to take some tumor cells and see if they're binding to the intact tumor cells, and seeing which of your antibodies from the serum are binding the tumor cells, or maybe light up the tumor cells and see if there are any B cells in that person's repertoire that are binding, that kind of thing.
Yeah.
That's about all I could think of. And and I'm not sure what Atreca has done, because they just got hits, and then of course they didn't really say too much beyond that, except for that they're seeing some clearance. It's a paper from a few years ago in, I think, Journal of Clinical Investigation.
Perfect. I will look it up.
Yeah, it's Daniel Emerling, Emerling, an active person in the Antibody Society.
Perfect.
All right.
Thanks very much. I think we can conclude now. So in concluding, I would like to thank Dr. Scott for relating her insights into the actions of the immune system. And thank you to our attendees for joining the webinar today. An on-demand version of it, as well as the on-demand version of a part one, which is already available. So that on-demand version will be available in a few days. I'll send a link by email to everybody who registered. I'll also include the link to part one. So please feel free to watch this or any of our on-demand webinars when convenient. Thanks again, and have a great rest of your day.
Thank you. Bye.
Bye. Thanks, guys.